All right, excellent. Jennifer, are you here?
Yes, I am.
Okay, great. Well, our next speaker directly from Spirit Lake by Mount Rainier. I don't know where you are, but it looks lovely.
It's a better
Jennifer,
view than my living room, so.
yeah. So Jennifer is Dr. Jennifer Hahn is a postdoc. I'll just say now in the Department of Entomology, but she, for the last few years, she's worked closely with us and has been in the Department of Plant Pathology. And she's going to be talking about Spore. What is it good for? Killing Varroa with fungi. I don't know if I said that right, but I figured you were trying to make some reference to that song.
Yeah, in my head.
Okay, go ahead, Jennifer.
All right. All right. So hopefully everyone's seeing my presentation now, yeah? Are we good?
Yes.
Okay. So first of all, I just want to thank everyone for coming today to this virtual seminar. So I'm going to be talking about some of the work that we've been doing with fungi to deal with this Varroa problem. Oops. There we go. So if you guys were all here this morning for Randy Oliver's talk, he talked a lot about Varroa and different methods for evaluating Varroa populations in your hives. So I'm going to talk a little bit about what Varroa is. If you guys don't know, it's a parasite of honeybees. Really bad thing, right? You can see the size of them on a honeybee. What they do is they're an ectoparasite, so they use their mouth parts to dig a hole more or less into the exoskeleton of the honeybee where they feed on the fat bodies. And this is very damaging to the honeybee. So this is a figure from a paper put out by Ramsey in 2019. And in it, they talk about how the Varroa feed on fat bodies, not hemolymph. For a long time, it was just assumed that the Varroa was feeding on hemolymph, and it wasn't really studied completely to figure out how accurate of a statement that was. And what Ramsey's group figured out was actually they're feeding on the fat bodies, and this is really detrimental to the honeybee for many reasons. First most, fat bodies play an important role in many functions of the honeybee, including immune function, pesticide detoxification, storage and synthesis of proteins and fats and other nutrients. It can help with wax production, metabolism, and thermal regulation. So these are all important for a honeybee to maintain proper functioning. So you can imagine a bee that has a bad Varroa infection can be more susceptible to the pesticides that it may encounter when it's going out foraging. So there's a lot of interactions. There's a lot of focus on Varroa as a vector for viruses, which is a really big problem. But it's also important to understand that Varroa can lead to a lot of other problems within the honeybee. So it can lead to improper thermal regulation. If it has issues with wax production, it might be able to be more susceptible to desiccation, right? It won't be able to keep the liquids in. So Varroas are really bad. So the question is, how do we control them? So for many years, beekeepers have been reliant on a lot of different chemical acaricides to deal with Varroa. But they've come and gone as resistance has been developed with Varroa against these chemicals. I mean, Coumaphos used to be a big chemical that's kind of out now, and Amitraz, there's a lot of reports of Varroa being resistant to Amitraz and it losing its efficacy. So it's really important for us to try to figure out another way of dealing with this Varroa. So what we've been doing in our lab and what my research has mostly been focusing on has been using mushrooms as a way of dealing with Varroa, as a biocontrol agent. So when I say mushrooms, typically people think of the mushrooms that you can see on the screen here, the edibles, right? Right now, my brain is full chanterelle mode right now because it's the fall and Hericium mode. But when I'm talking about the fungus that we use to deal with Varroa, I'm talking more about these kinds of fungus that you see here. These are from the cordyceps family. And these are the ones that are infamously known for infecting and killing different insects. So I think in common culture, you may have heard of the zombie ants. Those are infected by different cordyceps. Recently, not too long ago, there was a story about the salt shakers of death, which are cicadas that get infected by this fungus, where it hollows out the entire sexual organs of the males. So when they emerge, they get really excited and they try to mate with everything, but they have no organs. All they're doing is distributing spores. So these fungus can be incredibly potent killers of different insects. The one that we focused on in our lab was Metarhizium. So Metarhizium is the technical term, it's an entomopathogenic fungus. Basically a fungus that can infect and kill various insects. What's really important and unique about this fungus is it's already been EPA approved, safe for human contact and consumption, which makes it really nice as a treatment for honeybees, which is considered an agricultural crop, which means if it's safe for human contact and consumption, this could be a treatment that could possibly be applied during honey flow, which is, I know, a big problem with most other treatments that are available for Varroa. It's also been, other studies have shown that Metarhizium is not an effective killer of honeybees. And another really important thing about this fungus is that it's a native soil dwelling fungus. So I know that one of the biggest concerns about biological control agents is the release of non-natives into the environment and the impact it can have. And so what's really great is this is already there. So I was able to, I didn't use this particular strain, but I went to the local community garden plots here in Pullman, and I dug up some soil and I plated it out, and I was able to grow the fungus that I wanted, this Metarhizium, out of the soil in just a garden plot downtown. So again, it's all native, it's already there. We're not adding anything new. So it has a lot of really good potential. So we took this fungus and we did some initial studies where we tried to figure out, is this an effective control agent against Varroa? So we tested several strains in the hives. So we tested several conidiating strains, which is what this is. Conidiating meaning a strain that produces spores. We also tested strains that did not produce spores. And our treatment protocol was basically, we had a plate that was fully colonized by the fungus. We inverted it onto the hive. And we did this with 2 hives, or 2 plates per hive. At the end, what we found was that our treatment was working. However, there were some issues. And the biggest one comes from its thermal tolerance. So this was published by Rosalind James, where she looked at Metarhizium. This was years ago in 2009, and she found that the spores weren't able to survive in the hive temperature. So this is percent germination of spores at 4 degrees C, so your basic fridge temperature, nice and cool, and then 35 C, which is your typical hive temperature. And you can see that over time, and depending on how the spores are stored, but in general, at this typical hive temperature, the spores lose its viability fairly quickly. Some go down to 0% viability at 6 days. So this was a huge issue that really needed to be overcome. So we took on the task of trying to make our fungus more thermotolerant. So what we first did was we just looked at our spores. We kind of redid Rosalind's study just to see what we would get. And we stored spores at 4 degrees C, but I also did something where I tried to germinate them at either room temperature or at hive temperatures. And you can see spores stored at 4 degrees C at room temperature germinate fine. But when you're looking at hive temperatures, there is a real reduction in growth, meaning that they're really struggling. And if you look at spores stored at hive temperatures, and you grow them at room temperature, there's still this stunted growth compared to those ones that were stored in the fridge. And then when you try to germinate them at hive temperatures, after being stored at hive temperatures, again, the growth is really reduced. Saying that this fungus needs some help to be able to be efficient at the setting that we're gonna put it in. Because remember, this is a native soil dwelling fungus, and soil temperature is usually much, much cooler than air temperature. So what we did was we applied a directed evolution technique to induce to stress-induce conidia to develop thermal tolerance. There's a lot of literature out there that shows that fungus can be cross-protected for stress. If you induce, for example, osmotic stress, you can induce UV tolerance in addition to osmotic tolerance or thermal tolerance. So we went through two different stressors to try to generate a phenotype that we were a thermal tolerant phenotype. So we use osmotic stress and nutritional stress. Basically, we grew them up on media that had very little nutrients, the minimal media, so it had the basic minimum. And then osmotic stress, we applied it with a hydrogen peroxide treatment. And this is a general schematic of what we've done, where we where we induce some variation through our stressors, and then we grow them up at slightly above room temperature. We discard the ones that don't have the phenotypes we're looking for, and what we're looking for are ones that are able to grow quickly and produce enough spores quickly. And then we'll select those, replate those out, but we'll do it at increasingly higher temperatures until we end up with a strain of fungus that can grow at the temperatures that we are aiming for. And so we did that. And at the end, we got a strain that was thermal tolerant that we were happy with. But there's a lot of information showing that once you grow fungus outside of its host, it can lose some of that virulence, its ability to kill its host quickly. So we then did several rounds where we would take this fungus that we grew up in the lab, we would treat a hive with it, collect all of the mites that fell on the sticky boards, plate those mites out, figure out which ones were growing our fungus of interest, and then we would select for the ones that grew the fastest and killed the most Varroa per hive, grow those out, and then repeat the process over and over again until we ended up with a strain that we were quite happy with. And you can see, so these are images of the parental strain, the strain that we first started with, and you will see that these guys show that stunted growth that I had been talking about. But after our selection process, we have a significant change in morphology where this fungus is growing much better at our hive temperatures. So our goal so far has been pretty good. You can see over time, again, this gray line or these gray bars, you can see the percent germination of spores at 35C. And for the gray lines are the ones that were ungerminated. And the ungerminated, you can see, goes down by the end of the treatment, but the percent that we have that are germinating at our target temperature after selective rounds of treatment is much higher. So this is a video that I took, I think last year, of, so these are two Varroa that I have dipped in our spores, and then I plated them out. And you can see as they die, it's the fungus here that has colonized the body, and you can see it growing. So this is the mycelium as it grows. And as it's turning green, these are the spores that have developed, have completely taken over and killed this Varroa. So this is how this fungus works, is, let's see, go back to the binning. You can see these guys were just dipped in a spore solution. So the spores will grow what they call an infection peg. And it's amazing. It's just one cell thick, and it will literally drill into the exoskeleton of the Varroa and then distribute its hyphae inside, and from the inside proliferate and kill the Varroa from the inside out. And so that's what you're seeing here is as these legs curl up and die, they're getting desiccated. The fungus is growing inside, and you can see it's finally burst out of the body. So with this final strain, what we did was we went through a stationary berry where we treated it. We did that successive rounds of, we did these successive rounds of selections within this apiary where we were treating, growing up, selecting, treating over and over again. And we screened, I think, something like 30,000 Varroa to figure, to go through and make sure we're selecting for the right strains that we were looking for. And you can see over time, this is the percent mycosis of Varroa killed by our fungus. In that first generation, wasn't very high, but this was not unexpected because Varroa, as I mentioned earlier, when you grow them in vitro outside of their host, they tend to lose their virulence. But generation 2, after one passage through the host, it quickly regains it. And we can see that over time, the length of period, the duration with which our spores are infectious has increased, which is a really good thing. Again, this was our goal, to develop a more thermal tolerant strain. And the other thing is, this is a photo of, this is a spore print left on the top bars of the hive. And when I do a swab for these, I find still very much viable spores, which is a good thing. So not only do our, so this is a graph of the longevity of our colonies in this apiary treated with metarhizium or not treated. And you can see that the treated hives have an increased longevity compared to the untreated hives, which is fantastic. So the next thing we wanted to do was figure out, how do we get this as a mass production? So growing it up in petri dishes and treating hives is great for an experiment to do here in the lab. But as a actual application, it's a bit unwieldy as far as growing in bulk to make it a real viable solution. So the next step we did was we looked at different growth mediums. So metarhizium is a mold-like fungus, so it grows great on different grains, but it's a question of which grain is the best. So we tried birdseed, cob, oats, brown rice, wheat, and then we tried a bunch of different kinds of barley, because there's a barley processing plant just 10 minutes from here. So we did whole barley, pearl barley, and then compressed barley hulls. So I inoculated all of these guys, and you can see after just a few days of growth, I think this was less than a week, you can see that there's different colonization levels depending on which grain that we inoculated. Oat shows kind of the worst, whereas pearled barley and rice had the most promise. So when I fully let these guys go to conidiation and I counted the spores per grams of grain that we got, ended up, brown rice turned out to be our best growth medium for this fungus. So the next thing we did was we treated some hives with either oxalic acid, which is a typical Varroa treatment, or we treated them with 120 grams of our colonized rice grains. So these are rice grains, and we place these in a natural fiber. It's a banana fiber bag, so it's a natural fiber. We were worried about introducing anything too weird into the hive that might confound our results. So we went with this type of bag. We wanted to see how the bees would interact with it. It was really nice because the holes are small enough to keep the grains in, but big enough that the bees can still poke their heads through. And what we saw was that, you can see this photo here, the bees, so the way we treated this was we added a spacer between this top box, and there were some honey supers actually on top of here. And what we noticed was that the bees interacted with these bags perfectly. It's kind of what we were hoping for. You can see here, a lot of these bags look a little empty. So they've chewed holes through these bags, and they've opened them up, crawled through them, and distributed the grains throughout the hives themselves, which is what we're going for, because contact is crucial for this fungus to be effective. And so here are some of those treated bags I took out of the hive at the end of the study, and you can see there were quite a few bags where there was only a few grains left, and there are some bags that there was a few more, but all of them had been opened up and crawled through, which is again what we were looking for. And so here are kind of the results of this. And what's really promising here is that the metarhizium seemed to work just as good, if not maybe even a little better, on par with a typical oxalic acid treatment, which shows that, again, metarhizium is a viable option as an alternative Varroa treatment. So this was before treatment and after treatment. You can see this is what, 18 days later. And you can see typical for an oxalic acid, when you look at sticky board mite drops, there is a peak of drops and then it goes down. Whereas with our metarhizium treatment, we are getting continually increased mite drops, which is good because it's showing us that our spores are still working after, even after 3 weeks. So the other thing we wanted to do was we looked at using felt impregnated with our fungus to see if this was an alternative method for delivery. So the biggest problem with this method of delivery that we discovered was that for whatever reason, the bees did not like this spacer here between this and the honey super above it. And they had a really hard time crawling, making this 3-inch gap and finding those honey supers. So we looked at maybe felt strips as a way of delivering it. And so this was just a crude thing where you could wire it in and maybe replace a frame and instead have a frame of impregnated metarhizium. But before we went and did this in the hives, we decided to do a smaller scale study where we looked at metarhizium on felt strips in cages using bees and cups and an incubator. So this work was done in conjunction with an undergraduate student of ours, Riley Schultz. This was done last year and we're repeating it again this year. And so basically we had cups that were either no felt and fungus or a small, medium, or large strip of fungus. And we did longevity to see how long they would live. And we also looked at qPCR. So we harvested all the bees, we had them extract RNA, and we looked at different immune system genes and viral genes to see what was affected by our fungus. And what's really encouraging is that we also saw longevity in the metarhizium treated bees in cages compared to the control, similar to what we saw in our full-scale experiment with the hives. So this is another affirmation that even though we're not positive the mechanism with which the fungus may be increasing longevity, there seems to be a definite trend where bees treated with this survive longer than their counterparts without this fungal treatment. The other thing that we found really interesting is that, so dicer is a gene that's involved in the RNA degradation pathway. And this is really important in dealing with viral infections. We saw here control that there was kind of a spike and then the non-significant change between the first and last collection of the honeybees in this expression of this RNA degradation gene. However, what's really interesting is that there's definitely a trend you can see in these colored bars. These are all the cages that were treated with fungus, low amounts, medium amounts, and high amounts. And it gets more pronounced as you get with higher concentration. But what you can see is that the fungus, or the bees that are treated with this fungus, have higher expression of this RNA degradation gene, which implies that it may help rather indirectly with dealing with maybe viral infections. What's also interesting is, I'm only showing you the results we have for dicer, but we looked at other genes in the RNA degradation pathway, and we found the same sort of pattern in those genes as well. So another one I wanted to bring up is PPO. This is a known immunity protein. This is a function in melanization and encapsulation of foreign bodies in the bee. So this includes things like bacteria, fungi, and parasites. And we found here, again, in our non-fungal control, that there was a decrease in expression. And the same goes for the low and the medium amounts of fungus. However, in the high treatments of fungus, we see, again, I wouldn't say an increase, but maybe a maintaining this level of expression. What's interesting is, so this may be important in maybe because there is a fungal infection, but it also helps deal with perhaps parasites. So Varroa, it might help in the wound healing from Varroa feeding. So that might be really important there. And also there's been some studies showing that PPO is important in other aspects of bee biology, including the study by Shao in 2012, where they found that PPO in the hindgut of the honeybee is used to melanize pathogens in the feces and to eliminate those pathogens before they get excreted out. So this may be really important in helping the insect not reinfect maybe their food source with whatever pathogens that may be inside their body. So these are really interesting results. And like I said, right now we're redoing this study, but we're upping the concentration of fungus that we've used to see if we can find a more pronounced effect of some of these things. And then the final thing I'll quickly mention, because I had mentioned that we had done the comparison of metarhizium and oxalic acid and their efficacy and the ability to control Varroa mite populations. So metarhizium, the fungus, is known to produce calcium oxalate, which is very similar to oxalic acid. So it naturally produces oxalic acid. So we're wondering if there was a synergistic effect, and a study done by a master's student here you can see shows that there is a synergistic effect where here's oxalic acid and Varroa that were treated with a combination of spores and oxalic acid had the ability to control Varroa much better than oxalic acid alone. So we're going to look further on into the other synergistic effects and if we can treat with both oxalic acid and metarhizium to get an even better control method. And so with that, I'd like to open this up for questions.
Hey, Jennifer, it looks like there's a question. Okay, Laura's answering that question. So I don't know, I guess that's the question for here.
Oh no, I'm not answering it. I just made it so that Jennifer could see it. Can you guys all see?
Oh, okay, thank you.
Was there any mortality rate increase with the bees during this experiment? No, actually. We haven't seen any increase in bee mortality rate with any of these experiments, which coincides with previous research showing that this fungus had very low, if any, mortality at all on bees.
Jennifer, I have a question for you.
Yeah.
Did you use the felt in the hive eventually?
We have not done that in the hive yet.
I'm just curious how the bees' reaction to felt. I'm studying substrates for oxalic acid application, and I hadn't thought of using felt. Okay.
We haven't done it in the hive, but we have observed them in the cages, which is actually quite nice because you can see them much closer and how they interact with it.
Oh, yeah.
And it was really nice. They were quite happy to interact with the felt. We found them crawling all over it and behind it and all around it and exploring it. We think, at least for us purposes of contact, it's a decent alternative, or at least
it's a mammal hair, isn't it?
Well, so okay, I use synthetic felt.
Oh, okay, different thing.
So I don't actually know. I should have stated we use synthetic felt because we were autoclaving all of this stuff too, and we just kind of wanted to. But yeah, I mean, at least with synthetic felt, we just went to, I think, Hobby Lobby down the street.
Okay.
But yeah, they seem to like it fine.
So Randy, you don't have a need for wool, do you? Because if you want to make felt, I could send you some fleece.
No, you'll see tomorrow when I talk that I'm using different matrices for applying the oxalic acid and glycerin. I'm testing several right now. And I'm wide open to suggestions on those.
Yeah, so.
I was just going to say, if there aren't any other questions, I would suggest everyone come to Nick's talk later where he'll talk about the other half of the fungus that we're doing with bees.
So, Jennifer, I have a question. I mean, I think I know the answer, but what's the, what do you think right now your best guess of what the sort of substrate that would be ideal would be? I mean, could you not use those banana bags, but make them thinner where they just fit between
Yeah.
two boxes?
Yeah, that's kind of what we're thinking about doing next. Because it really is just that spacers seem to provide, for whatever reason, in the minds of the bees, they didn't see that as still part of their hive. And so they had a hard time going up into the honey supers. Yeah, I think if we could lay them flat, we even talked about doing like the cover boards maybe that people use in the wintertime to put their sugars on. Something like that. Yeah.
So Jennifer, how many generations have you tried running on the rice grains? I'm wondering if it will lose its virulence towards Varroa when you start culturing it on rice grains.
That is actually something that I had considered, and I hadn't mentioned this, but the other thing that we're doing is I amended some of the rice with chitin.
Okay.
To kind of help keep that virulence up as part of a natural substrate that it might encounter.
Right.
But the other thing is,
so let me ask you this.
It's an ongoing selection process.
Did you use shrimp chitin or insect chitin?
I use chitin from Sigma.
Oh, okay. Now, the reason I'm suggesting this is realize, because I'm doing experiments with oxalic acid and chitin also. Yeah. And shrimp chitin contains calcium carbonate, which very rapidly neutralizes the oxalic acid. So, so you may want to, if you're going to use it, make sure you don't get a shrimp chitin, get an insect chitin.
Yeah. Well, the other thing is, I don't know how much people are interested in it. I'm using colloidal chitin, so it's chitin, but you have to process it. So we spin it in hydrochloric acid, and we do a bunch of different washes to get it out to a different form.
Okay.
So I'm using a purified colloidal chitin as an amendment to the grains.
Yeah, I'm also interested when you start culturing the metarhizium too, specifically for Varroa, for the temperature and for Varroa, you're going to want to keep also running your mortality tests for the honeybees to make sure you're not co-evolving it to be more effective on the honeybees.
Yeah, exactly.
Yeah. I mean, you with Christine Peng's research with Hirschutella, where she did much the same thing way back in the years ago. Okay.
Yeah. So I mean, we focus on metarhizium, but Hirschutella, Bovaria, there's verticillium, there's quite a few other entomopathogenic fungus that people have explored.
So she Jennifer, did the passaging through Varroa also, and did the selective breeding also on that, and I thought that was very clever back then. So you're doing the same thing. Good way to go.
Right, and the other, I think, Jennifer, that I think you made it clear, but it would have to be an ongoing selective process to keep the virulence up, right?
Yeah, the selective process up.
Because we want to keep that. We'll survive that Varroa.
Yeah.
It's
weird for some of our experiments trying to collect live Varroa. But what you were saying before about that powder shaking method, the powder roll, we found very perfect for our ability to collect live Varroa.
Now, you don't need live Varroa for culturing. Could you culture it on freeze-dried Varroa?
Oh, well, this is for a different experiment where we're looking at the synergistic effects of the oxalic acid with metarhizium.
Right. I'm just talking about maintaining your production of the strain on Varroa.
No, I don't think they would have to be live.
You think so? Oh, okay. Because you think so having the rice grains alive, is that critical?
Having the rice grains alive? No. Having the Varroa alive is not necessarily important for its infectiousness as long as we have that Varroa body to grow on.
Right, so you could use dead Varroa.
Yes.
Dead Varroa are much easier to harvest than doing powdered sugar shake.
Oh, I know, but for an experiment where we needed live.
Oh, no, I'm talking about production, the next step up, ramping up.
Yeah.
Okay.
Okay, well, if there are no more questions,